Francisco Javier Sobero is currently General Director of the National Institute of Genomic Medicine and a Senior Research Scientist of the Biotechnology Institute of UNAM, the National University of Mexico. He holds a PhD in biomedical research from UNAM. His research has focused on the chemical synthesis of DNA and its applications in the study of proteins, as well as the development of biopharmaceutical and vaccines and biocatalysis. More recently, he has focused on genomics and its applications to human health. He has published more than 70 original research articles in journals of international circulation. Start, please.
Thank you. Well, I'm very glad to be here. I enjoy very much the opportunity to people from different approaches to knowledge, and especially being in an environment where there's academic research of from different perspectives. It's particularly attractive for me. And I'm also very enthusiastic about what's happening in the area of the biological sciences, and especially this buzzword that is genomics, which is genetics of the 21st century, let's say. And I will try to do a little bit of explaining why is it that genomics is becoming a useful tool for studying many things, including human behavior. So I will refer to basically all the concepts that are listed there, not necessarily in a particular order. I will go a little back and forth. But I will speak about the genomics revolution, the relationship of genes and human emotions, and in general about the biological basis of evolutionary behavior. So I will start with some basic tenets, at least are basic tenets for me, maybe from some of you as well. The first one is this phrase that is known from other contexts, and a molecular biologist Nobel Prize says, when referring to the nervous system, he says, no matter, never mind. And it's clear that the mind today is understood as something that has a biological basis and a biological manifestation. It's in the neurons, right? So, and I quoted this, I also brought this from a neurosurgeon that says the idea that my tool is moving through thought itself, through emotion and reason, that memories, dreams, and reflections should consist of jelly, is simply too strange to understand. Nevertheless, it is a fact. The other tenet is humans are a product of evolution. We are here due to a process. And of course, Darwin, much facing some trouble, probably, stated that in one of his books after the origin of the species. And I think the last part is what I want to emphasize. Man still bears in his bodily frame the indelible stamp of his lowly origin. So we are a product of evolution. That's a basic tenet also. And this one is probably a little bit more, I guess, I don't know if it's less obvious, but sometimes I find it not as shared as an idea. That is, a human brain is a direct product of a human genome. A human genome produces a human brain, and it's different from a chimp's brain, and it's different from a rat brain. And it's already, since birth and before, it is already a human brain. So it's biologically predisposed. It has some inherent capabilities and inherent tendencies to operate in a particular manner. And that's also a product of evolution, of course. But then we can understand, and the human brain is determined by the human genome. The structure, the basic structure of the brain, it is a direct product of the manifestation of the genes. It It hasn't been influenced, maybe in a very subtle and marginal way, but basically it's a product of the genes. So we can understand much of how it works by understanding the genetic code that produces it. Yes?
So there's a question. When you say it's a direct product of the human genome, you don't want to say independent of its environment, right? I mean, it is that it's the interaction between the environment and the genome. It's the human brain.
Well, the human brain itself, the way it is being formed in the womb starts to receive some environmental cues. Of course, that I understand, if that's what you're saying. And of course, immediately afterwards, and especially after birth, it starts receiving a lot of content, and it starts accommodating a lot of content. But that it requires to be a human brain, and it already is a human brain because of the genes that instructed the cells to produce that. That's what I'm trying to say. I'm not, of course, saying that the environment has nothing to do. Of course, that's most of what we are is a product of the environment, of course. So this is another. It's not a basic tenet. It's just that we know some things about the basic, some even strange things about emotions from natural experiments. This one very famous is this accident that happened to this person, Phineas or Phineas Gage, that received this steel bar crossed his brain, and he raised from the accident on his own, and started speaking again, and it seemed fairly normal. And actually, after some time, it was determined that his faculties were basically not much perturbed, but he lost all control of emotions, and he was the foreman of the crew, and he became completely socially unadapted because of the perturbance of that part of his brain, which is more related, it has been seen with planning, moral judgment, and emotional control. So, of course, there is evidence that certain areas of the brain are directly related with emotional responses. This one other, I haven't found. I know I read this in Time magazine maybe 30 years ago, talking about the neurosurgeons. But this, I remember it very well. This stockbroker who lost his successful judgment after brain surgery, and all the faculties intact, intelligence and number capability and reading ability, everything, except he no longer felt fear or when entertaining risky transactions, and he became a failure just because his emotions were not in tune with his activity. And I found that extremely interesting. Well, I think none of that what I said is new. Everybody knows that, or to some extent. I think it's relatively easy to be more or less in agreement with some of those tenets or concepts. But what is new that I want to bring here today is that now genes are accessible. Genes were fairly inaccessible, very indirectly we could get to them. But by virtue of the genomics revolution that started in the last decade of last century and has started with a very impetuous pace in this century, we now have access to the genes. Why is that? Well, just a few things to say. From the discovery of the double helix, from the discovery that the material of heredity is a chemical strand which has a code with 4 chemical letters, to the possibility of starting the study of a whole genome of a human. 60 years elapsed, And this is what we call especially the molecular biology era, and then the recombinant DNA at the end of the century. But then the scientific community there start to think about sequencing or obtaining the code of the whole genome. This was unthinkable even in 1985 when I learned for the first time we're planning to do it. And I thought it was completely nuts, impossible, because of the power at that time. Well, sometimes I can't resist but saying that I did the first DNA sequencing that was done in this country, 1979, and it took me about 3 months of hard work to sequence 150 letters from my genetic system. And then I was told 10 years later, or less than 10 years later, that they were going to try to sequence 3.2 billion letters. How was that going to be possible? Well, in fact, it was possible. Technology advanced. And then in the last 15 years, these are the front pages of the different the journal Nature, which featured a particular genomic advance. And there are many different areas where it started, where it impacted having human genome. This has come to a situation right now that is, I think, really astonishing. This here is the very well-known Moore's law of the advance or the progress of computing power per unit of cost. And this is, of course, it's exponential or logarithmic scale. And this was the cost of sequencing genomes or sequencing DNA, the basic technology for genetics. And this is what happened about the middle of the past decade, about 2006, 2007. And it was a major technological advance. And so we have now a technology that has dropped in cost 1 million times from the time that the human genome was first deciphered. So it's nothing in the history of mankind, no technology has dropped in price as dramatically as this one. It used to be the time when we measure one letter at a time in an experimental system, and now we have these chips which can measure 5 million in a few minutes, 5 million positions of a genome in just a few minutes with very, very highly reliable results. And that's why the first genome cost $3.2 billion, around $1 a base. And then at this time, we are in $1,000 per genome, per new genome. If any one of us wants to decipher his own genome or her own genome, will cost $1,000. And we are approaching the point where we will have 1 million human genomes sequenced around the world. So we have access to amounts of data that were unimaginable just a few years ago. So that's what the genomic technology has advanced. And it opens several possibilities. One of them relates to the evolution of the human species. Genomics opens a history book. So we now have, I think mainly because of the genomic comparisons of the genomes of all these species and many others, I think it's firmly established that the divergence from the orangutan happened in the common ancestor of humans about 14 million years ago, with the gorilla about 8 or so, and with the chimpanzee it's about 5 to 7 million years. We know there are two types of chimpanzees, 2.5 million years between one of them, between the two varieties of chimpanzees, which are our closest biological cousins, extant cousins, of course, and the human related to all of them. And there are Of course, what we now distinguish hominids, these are all hominids, from hominins, which are the ones that are after the chimpanzee. So the evolution of these is mainly on the fossil record, because our closest cousin is the chimpanzee. But we know from the fossil record, and now we know from other sources several of these intermediate varieties of hominins. Okay, the earliest until the Australopithecines, and check here up until 3 million years ago is about when the bipedalism started, and several other things were happening. And Homo is about 1 million years or so. These are two or one, between 1 and 2 million years ago. So, but now we have a possibility to access these states, because we can check the differences in mutations of individuals that we sequence today. And then we make counts, and we take what we the biological clock or the genetic clock, and we see so many mutations imply so many years, or so many millennia, or so many million years. And further, we have been starting, scientific community has been able to sequence ancient DNA that has been fossilized. I think one of the most amazing possibilities is to sequence DNA from hundreds of thousands of years ago, and we have now the complete genome of several Neanderthals. Okay, and these are taken from remains of the bones. This one is really interesting, I think. This is the piece of bone that was used to find not a Neanderthal and not modern human, but also living in Europe, which is called the Denisova, because it was found in a cave. And because we were able to sequence the DNA, we know that it's not a Neanderthal, it's not a modern human, and we also know that some of the genes of that species or that subspecies is present today in some populations in the world, especially in South Asia, only there. But we don't have a skeleton for this species. But we already know it was not a Neanderthal, not a modern human. So that's what has been possible to do with the new technologies of DNA sequencing. As you can see here, we know this is some biological group, okay, coming from several million years ago, separated about 600,000 years ago from the Neanderthals, and then the Denisovans separated from them. And all this we know because we can access their DNA. Lots of the DNA. In one case, the whole genome. Well, so we can now take a look at human evolution from a completely different perspective, because we have access to all those data. Now, a little bit about how we understand emotions, or the way I understand emotions, being just a natural scientist. In its relationship with the brain. So we know that basically there's a subset of the brain that is more specialized in housing emotions, although everything is communicated. We have this limbic system that we share with other species that are, similar to us, mammals and primates. And there's other parts of the brain that are much more primeval, much more related, shared with species that have diverged lots of time ago. And then the cortex, or the neocortex, that is probably only, was present certainly in some of the other, like the Neanderthals or the Denisovans, but not in other mammals, and not in the same way in the chimpanzees, for instance. So I think that's why being emotions from that perspective has, I think, given good health to this particular approach that is called evolutionary psychology. I find it really interesting. For instance, some new things that are happening from the biological perspective is recognition of this concept of evolutionary linguistics. So trying to understand that language is something that also evolved, and it evolved with some of the human and pre-human lineages. How it did, it was not a valiant endeavor until the very recently, I learned. And also the importance of the control of fire. It's probably over 1 million years ago that humans started controlling fire. And that changed completely their social relationships and started to also to mark some differences between the behavior of males and females. And it's there. Of course, it modified completely our digestive system, but there are signals of that progress, of that evolution of 1 million years, which is a significant time, which has been marked in the genes. Well, it's interesting again that Charles Darwin said this in the Origin of Species. He said the discovery of fire, probably the greatest ever made by man, excepting language, dates from before the dawn of history. So we, according to this view, we are beings that have been modeled by evolution for sufficiently long time to distinguish these effects in our social and emotional behavior. And that's not possible. It is possible now to start to dismember it and to analyze it more systematically. The other thing I want to convey to you is I think it's very, very interesting, very useful to see that evolutionarily humans have been in what we call the cognitive niche. I think it's a useful concept. The cognitive niche, look, what says is that humans have created a new biological niche. And that niche is a place where humans that become more intelligent and more socially functional leave more descendants. And so that's why this niche, it didn't exist. It was even created by human societies. And so that's the context where we should look at to understand how human evolution works. Technology is there for over 1 million years or more. So this technology is where humans have to thrive using technology. The individual that doesn't learn how to use technology will die. So that's sort of the idea. Of course, having language. It's very important for survival to be able to use language. So there are pressures to maintain and even to enhance linguistic capabilities in these populations that were evolving. And other things like cooperation among non-kin, very human, and very related to emotions. These are things that evolve in these environments. Some of other human characteristics are there. Of course, human division into cultures that differ in language, customs, diets, depends extremely importantly in cultural or learned information. What I'm saying is there is a substrate that is amenable for study that is in there, and we shouldn't ignore it. I think it's extremely, I think it will be extremely capable of explaining much of what we humans are. And it does relate to particular genes. We are now starting to be able to discover which genes are doing what. Some of them are, I think it's very, very interesting to look at. This one I like, this Fuzz B2 mutation, pronounced in Spanish. It sounds even more provocative. But it's related to language. And the mutation it is possible to say that there is a mutation there that has adapted value. Technically, we can say it because of the nature of the mutation. And so we know that there is a mutation that was fixed in there, and it promotes a better use of, we know it's related to language because families which have the mutations in that gene are impaired in their language abilities. And we also have been able to start testing it. If you give a mouse the human version of FOXP2, you will have a mouse that cries differently, and also a mouse that has some interesting nervous system development, which changes. This is a control gene. It's a gene that controls several functions, and its function in the brain is, of course, not very well understood, but it has relationship with communication and language. A similar one is this. I will talk a little bit more about that one, this SRGAP2 gene, a duplication, duplicated in hominins, not in the other hominids. So we can start mapping some genes and some functions to particular moments in evolution, because we can check which of our related species have the versions or not have the versions. I will do a little bit more of propose a few more examples. So you see here some of the behavioral and cultural characteristics and the relationship that they may have with particular emergence of gene variations. Okay. For instance, we know that Neanderthals already had this FOXP2 mutation. One thing I found astonishing is that in the mainstream of the linguist community, they assume that the language is something that emerged about 70 to 100,000 years ago. Genetically, that's absolutely untenable. It has to have evolved at least 250,000 years ago, which is the last common ancestor of all humans that live today. And every human is capable of acquiring language indistinguishable from every other, unless it's sick. But that's not a population related. But these things we learned just recently. So we know that fact we know because of recent finding. A few examples, I think, very, very provocative, at least for me. This gene, it is certainly involved in nervous system development, brain development, and chimps and orangutans have only one copy. Every human being has between 4 and 6 copies, and not all have the same number of copies. It's something to think about. Okay, this is very, very likely related to the events that were at the very start of the hominin lineage. We now know other things. This is another very interesting gene. It is also a control gene called HER5. The human version, you give it to a mouse, and then you get expression at some time in development. So this is the chimp version. And then if you give the human version, it starts producing its products earlier, and it promotes a larger brain. Okay, and this version is different from the other hominids. So we now have a few candidates for the crucial differences. So we know we are different from a chimpanzee on average about 30 million positions of the genome. We are different from each other about 3.5 million positions, and we are 10 times as different from a chimpanzee. So which of those 30 million modifications are important? Seems hard, and it is hard, but it's becoming more and more amenable to study. It's also maybe some of you are questioning, how can we make a mouse with a human gene? And we can do that now in a fairly standard way. So we can modify and give those genes and experiment in some of these animal models very well. We cannot do it with humans, of course. And this is another, coming to the conclusion, the hominin lineage, it's very interesting and it's very different in some regards that they pertain to the dynamics of the genome. It turns out that our genome, each bar here represents one of our chromosomes. So we have the sequence of all of it. And we know that there are many repetitive positions in there, remains of viruses and other things like that. And it turns out that that promotes that recombination between some parts of the genome and others occur much more frequently than even in other hominids. Okay? We recombine our genome, our lineage recombines the genome more often than the chimp and the gorilla, which is fairly surprising because we are so close. So it seems that part of human evolution, it has been the result of a particular dynamics. And it turns out as well that some of the psychiatric diseases occur frequently because of these dynamics. So it seems that our genome has been a producer of diversity related to the nervous system and to the brain at the cost of producing people that are mentally ill. It's a very, very, I would say, scary concept, and it's still preliminary, but I think it has some data supporting it. Okay, so behavioral traits and the appearance of mental illness seem to be related because our genome is inventive in creating brain modifications. Okay. I don't know if this was too technical. I hope I made clear what the implications are. Now, of course, there are other genes, not necessarily from an evolutionary perspective, but directly related to emotions. And we know that there are several products of those genes that are clearly related to emotions: dopamine, oxytocin, vasopressin, serotonin. These peptides are some other substances that are clearly related to certain areas of the brain, and they turn on and off, and they operate in the nervous system in different moments, and of course reacting to cues from the environment. And I will cite some of the ones that are the studies that I find more provocative. For instance, we know oxytocin. This is the hormone of empathy, the hormone of, let's say, in certain way it could be called the hormone of love. Oxytocin in the brain creates reactions that are very strong. In the case of rats, it has been studied, and this is very nice because it shows how a pre-existing network of interactions reacts to an environmental cue. In this case, the pups crying. When the pups cry, they turn on oxytocin in certain areas of the rat's brain, and then the mother becomes a mom. It says so now it turns on the behavior to take care of the pups. So there is that gene product, oxytocin, that is related to behavior, that is related also to emotions. This is the one that I find most provocative. This Fuzz B gene, which is related with nurturing. When there's mutations in this gene, in the female, they are absolutely normal mice. in every regard, except after they give birth. After they give birth, they just don't start the nurturing behavior. And then when we check the brains of those rats, it never turns on the program in the neurons. And that's why these are bad mothers. This is their only defect. They are bad mothers. Now, the question, this very, I think it's obligatory question. Do we have a Fuzz B? Or do we humans have Fuzz B? Does it do the same? Well, nobody has dared to study that, but we could. We certainly could check what is the different versions of Fuzz B, and if they are related to particular behaviors after birth in women. It could be studied. It hasn't been done. I've been checking the literature repeatedly. Now I will resort to talking with the postdoc that did this 20 years ago, but nobody has studied that yet. But of course, I have to refer here to the other enormously important component that we can study by studying genes, and that's epigenetic signals. The signals that come on top of the genes and that are a reflection of the history of those cells, of those individuals, and that become imprinted in the structure of the genome, but not in the code. Okay, it's like the chapter of a book. The chapter is identical, but I have a clip and I have it silent. Nobody can read it. Or I have the book open already and I pass along the book already open in particular chapters on particular places. So it's just turning on or off certain parts of the genome on a stable basis, okay? And it can even skip a generation or two, some of these signals. And these signals, some of the best characterized also in rats, is related also to mothering, but also to the effects of good mothering or bad mothering. It says good mothering in there. It seems that Mexican mothers don't do too much. Here, and this is the neglecting mother. Okay, there's ways to make rats behave that way, but then when you study the genes, you know you can very clearly and repeatedly. It's very very well established fact that when maternal care is high, you generate an active receptor for glucocorticoids, which are the stress hormones. And then these genes remain active for the life of the rat. Whereas when there is neglect, these genes turn off and remain off for the remainder of the life of that pup. So this is an epigenetic effect, which is durable, is not inheritable. So these, when they become mothers, they will reset the state of a gene. But it didn't change the code, it only turned it off. But the effect is very important in the future of the pup. We suspect that this is very much similar in humans, and that much of experiences, especially early experiences or prenatal experiences, are imprinted in our genomes, and that's a whole other dimension, even at the genetic level, the epigenetic level. Of course, needless to say, all the other effects, social, cultural, educational, and so on, which are dominant in the human as a species. Now, how do we study that in humans? We cannot make humans knock out the genes. We cannot, open their brains when they're alive and see. Well, we can use imaging technologies like the ones that were already described and are becoming more and more useful. But we can also do these types of studies, what we call genome-wide association studies. You take a set of individuals, could be humans, which have a certain characteristic. You try to isolate that, as a variable, and then check their genotype or their genomes. And then take another set, which is different. Let's say, for the sake of my previous imagine experiment, mothers that abandon their children and mothers that are just normal mothers. And then you scan the whole genome, the 32 billion bases, and say, in which positions they are statistically different. And that pinpoints where a gene that is related to that particular trait is located. And we can do that, especially now that we have 1 million genomes, that we can spend $100 and then have the genotype of any person. We can do these studies at our heart's content. It's just a matter of imagining the experiments and the studies, and of course obtaining the informed consent of the individuals. And the other is candidate gene analysis, inspired by animal and other type of studies. So we can do that as well. So it is open for study. Some of the recent discoveries are, I think, very thought-provoking. Genes related to aggressiveness, very, very strongly related to aggressiveness. And in this case of the monoamino oxidase, we know also the epigenetic component. Certain mutations generate almost always very aggressive people. 90% of them go to jail. And some versions of this same gene are only make people prone to aggressiveness, but a more favorable environment makes them, the manifestation of that version of the gene is much lighter. And so there are many others. I like this one, very recent, the genetic basis of music ability. That's very related to emotions. And there are some genes and some areas of the genome that are related to that. And this is last week, Genomewide association study of loneliness. So they relate some genes with people that feel lonely, and there's a signal. By the way, this signal here, this is log P, so it's very significant. Not necessarily has a strong effect, but undoubtedly influences that trait. That is very sure. In this case, loneliness is just genes related to loneliness, but not much more. It could be very small effect. Just to end up, this is oxytocin methylation as measured in saliva, and display and makes it possible to differentiate some more secure attachment style from others, and its capacity to recognize emotional facial expressions, depending on the variety of the gene that the people inherited. And so, to me, this perspective is a few genes have been studied today, but the new capacities of the technology, it really opened the possibility of studying many, many, many more. And that's not to say that we will be able to explain by genetics a lot of human behavior. Only a fraction of human behavior will be related more directly to genes. But that fraction I find fascinating. Thank you.
Thank you very much. The discussion is open.
That's your job.
Tú me dices a quién.
Over there. The difference between genomic in humans and chimpanzees makes some useless the study and homology between cognition and behavior in, for example, bonobos and human, with the goal to explain moral or empathy or cooperation?
Of course, I ponder this question as well. If you see the times of divergence, so in 2.5 million years, bonobos and regular chimpanzees, I don't know how to call them, Pampaniscus and the other, Pantroglodites is the name, very apt name for the one that is more aggressive. Their sexual behavior is completely different. It's only 2.5 million years. And so if you see that, you see, well, what is the relevance of their behavior to ours? 6 million years divergence. I still think that we can learn, if we are wise enough, we can learn even from rats or from bees or ants. We can learn, only we have to be sufficiently, clear in our hypothesis on what is translatable or not. But I think the caution that I understand in your question, I would agree with it. We have to be very cautious as to what is the implications of the behaviors of some of these species that are close to us.
Okay. Thanks very much for the talk. I certainly share the enthusiasm for this amazing science of genomics. I was a little concerned that in the talk you sometimes presented some of the particular genes you talked about, a neurotransmitter in one case, as if they had a single impact. And you used that language. Fuzz B, you said, only causes maternal deficits.
I may have implied
mice.
it, but I didn't say it.
You actually said it. Just correction just to make sure everyone is clear. Other studies show that it causes depressive behavior. It also causes epilepsy and some neurodegenesis. So
in rats?
In mice.
In mice. I'm sorry.
FOXP2, as you mentioned, is found in other mammals. Is also found in fish. The idea that it's a language gene has been popularized by the press, but people in your field, as you know, it's contributing something to language, but exactly what is a bit unclear. Oxytocin has been implicated in a number of social behaviors, but also in aggression. And its actual impact on emotion is, again, going to be quite varied and complex in ways that we don't fully understand. MAOA, maybe I don't know the new work that you mentioned with the 90%, but the original work on MOMA, I think, was really very effectively criticized. It's found in 30% of men. Its contribution to violence seems to depend on environmental factors. So I think you did, hedge and make this clear in some points, but I just want to make sure, and maybe to ask you whether sometimes the telling of the story gives the impression that there's a gene for a specific behavior. So maybe a direct way to ask you would be to say, do you think that there's any evidence that there's a gene for something behavioral?
Yeah. No, of course, I think you're absolutely right. And I'm very glad that you made the distinction. Every gene manifestation is what we call epistatic. Okay? So genes don't only do one thing. Most of the genes do different things. They participate in different processes. And it's only in some instances and some versions of the genes that have what we call high penetrance. I will barely touch a little bit in a few of the things that you pointed out. First, in the case of the mouse, this knockout, it is. I did say that it was normal in every other regard. Of course, I haven't taken it to the theater. I don't know exactly how it will react there. I don't know if it's already afraid of cats or no longer. So there are many things that we didn't try, or they didn't try. Okay? In the cage, in the obvious behavior, and that's part of a story. The postdoc thought that knockout was of no consequence whatsoever. It was of no consequence at the level of her studies. Of course, Fuzz B expresses in many other tissues. So it's only that it was possible for the mouse to supplement or to substitute for the absence of Fuzz B in most other regards, except in this one. But epistatic effects are very likely in each and every gene. Now, the language. It's true that they said that this is the language gene. That's ridiculous. It's impossible that a complex trait as the language depends on 1 or even 100 genes. It depends on many, many genes. But there are certainly some mutations or some innovations at the genetic level that we can definitely trace to crucial moments. And I think Fuzz B is going to be one of those. Of course, it only provides language in the context of all the other changes that were happening, okay? But of course, the complexity that you imply in your question and your comment and your observation, I agree completely with it. I agree completely with it. There is, now, monoamino oxidase, MOA. For each gene in a human genome, there are many alleles, right? Many varieties. Some of them have particular functions. We call it penetrance. And some of the mutations in monoamino oxidase gene are extremely penetrant in creating an aggressive behavior. If you inherit one of those, it's very difficult for a person to remain calm and to not be extremely aggressive. And that's what I say. Maybe I'm mistaken. I will check my numbers. But if you inherit that particular mutation and that you are homozygous for that, 90% chance you go to jail in your lifetime.
You don't recall the author?
No, I will check it for you. Yeah, I'll give it to you.
Thanks. Probably about 80% of what I wanted to actually say has already been. It's more or less the same point, but yeah, I'll try and push it a little bit further. What would be the state of the art in that sense of, let's call it multi-gene, multi-locus studies? I mean, one of my, obviously you've got to have an admiration and a fascination for everything that's been going on in the last 20 years. But the trouble is, it's a bit like the old thing with the drunk losing his keys at night, looks for them under the light, single snip things associated with very, very recognizable phenotypically, in ailments or diseases is so much easier than trying to get in among all the noise to look at multilocus traits. Where are we with actually being able to identify, because all the thing with behavior, as you say, it has to be multilocus, right? In general.
Right.
So where are we with, actually being able to have technologies that can actually be able to bring out multi-locus effects as opposed to single locus effects?
Yes, I think that's also a very, very good question. Thank you for asking it. One approach that is being called GPS. I always remember exactly what it means, but because calling it GPS was very, attractive. But it's something like genomic. No, it's genome-wide polygenic score. So that one takes, without any prejudice, all the traits. I mean, all the whole genotype, genome-wide, 1 million positions, and then you try to correlate the 1 million positions with the trait. And when you consider 20, 30 genes, you explain an important percentage of variability. Recently, I attended a conference where this approach was used, and they claimed that they were explaining about 30% of the variance of scholar achievement. This is a last year study, because that's one very easy to study according to social sciences. I learned because it's very easy. What is the maximum level of scholarity that you achieve? And using this GPS, they explain a sizable percentage of variance using many genes. No clue as to what the mechanisms are. Whereas when you have a candidate gene, monoamino oxidase or Fuzz B, then you can probably understand a little bit of what's happening. But the GPS is also an approach. And the other component to your question, I think, is we can now approach this using very large numbers of people, because the genetic component is immediately accessible for a very reasonable amount of money.
Sure. Just mention quickly why I have a problem with that. So So it's basically the following. So the thing is that you take as your class basically scholarly achievement, right? And then you look as your predictor variables, all these different genes, etc. But unless you put in all the other potential social factors like socioeconomic status, etc., you have no idea whether that set of genetic. So in other words, this 30% is really fictional in one sense, right? Because it's only if you put in all the other factors and you really try to disentangle one from the other, you'll be able to say, well, really it's a proxy for something else, which is really determining the scalarity. Right? You see what I'm saying with the...
Sure. I am among philosophers. So, undoubtedly, I think 30% of variance explained by the genomic component, it's only in that set of circumstances. If you change the set of circumstances, you may explain more or you may explain less of the variance. Because in some context, genetics is going to be very important. In other contexts, it's going to be almost irrelevant. So that is certainly part of the complexities. And as a scientist and as a molecular scientist, as a chemist that I am in the origin, I like to understand mechanisms, not just correlations. But I think we have now the light. Well, it used to be like a very dim candle. Now it's a strong light, but very limited. I think we will have. Pardon me?
It's still a correlation.
Sure. We are still in the stage of phenomenology, just correlations, and in some cases we start looking at mechanisms and possible interventions. Okay, which is, I think, some of the most interesting possibilities to start using what becomes knowledge, not data, but knowledge, and start to be able to intervene in a useful, constructive way.
We're back to one mic, so.
Thank you very much. I would like to ask you about the interest of the epigenetic effect. So the interaction with the environment has an effect in behavior. That behavior can go through memory, maybe changing epigenetics. So there is a very interesting part, which is we want to understand the mechanism. So, but I would like to ask you, beyond the way in which the interaction interacts with the environment, what do you think that is the interest of knowing which processes, which behavioral changes are mediated by epigenetics, for example? Do you think that they are irreversible? So what is the difference? So mechanisms aside, so it's clear that it's interesting just to understand the mechanism. So what is the difference between the relation between my mother, with my mother who was a bad or a good mother, having an influence into my genes and having an influence into my psychology?
I think the answer that you give is the one that I will subscribe. So it is different because one thing is more reversible than the other. Probably the methods that you will use to alleviate suffering or to promote a more constructive social person is different if you know that these are epigenetic effects that are in a sense I'm not saying they are irreversible, but the way to make them different is probably very different than the ones that are in your nervous system, as neural circuits. So the plasticity of a neural circuit is inherently different than the plasticity of a turn-off gene at the level of the histones and all that. So probably here the mechanism, it's important because it modifies. Maybe one thing, you can do some things with drugs and others you can't. So understanding the intimacies, I think it will be useful.
Is there any evidence of reversion?
Yeah, of course. What we call the programmed stem cells. It's an exercise so that you can take a cell that has become a skin, for instance. Lots of genes are there, turned on and off, and then you give 5 genes, 5 control genes, and after a while, this produces a Nobel Prize, you generate what is called induced pluripotent stem cells. So you deprogram them. Now, doing that with your nerve cells, your brain cells, it's another matter. But, you can conceive making some of these reversible.
Well, no, I don't know.
All right.
This is just a question about something you said. I was wondering how you mentioned the cognitive niche and how the constant reorganization of our genes generates some psychiatric illnesses. I was wondering how that works exactly.
Yeah, well, it has only some I don't know. There's a causal relationship with the cognitive niche. The cognitive niche itself is a product of the first manifestations of human intelligence, tool capability, and the consequential social modification of social interactions. Then it creates a new niche, which was not natural. So, and now for an individual that belongs to that new community or that community, now his or her survival depends on being able to progress in that created, niche. So that's what is the cognitive niche. It's like the beavers create a niche because they make the dams. Okay, and now the beavers, to progress, they need to learn how, and their genes evolve in a created niche. Now, in the case of humans, we created the cognitive niche, which is a niche in which only the progress depends on the capability of understanding things, of making better tools, of reading the other's faces and having an emotional relationship. That's what it's called the cognitive niche. Now, the other thing is, very surprisingly, the human genome has dynamics on it of all its own, which are different from even our closest relatives. Why is that? I don't know. Is that responsible for generating species with the very, unique characteristics? It's been, 400 million years since there are animals, and 100 million years after the dinosaurs, and it's only one species that has evolved these unique capabilities. Now, studying the genome is very provocative to see that our genome is also unique in the sense that it recombines and generates duplications at a higher pace than others. We don't exactly understand what is happening, but we also understand that some of the mental illnesses are products of these rearrangements, not of conventional mutations. So we generate mentally ill members of our species at a higher rate than expected by simple mutations, okay? And some of these rearrangements of the genome create modifications on the neural nervous system. That's very, it's surprising.
Related to this cognitive niche, I was thinking, is there any theory or some way to explain, because the cognitive niche could be first very primary or basic, no? Then it has been developed and there are different skills or capabilities within it. For example, evaluation and maybe even moral or thinking and just getting information as a receptor, processing, I don't know. But what I want to know is if there is any research which relates these different capabilities to certain the names of these genetic parts, I don't know, I don't understand very well, or is it more general?
I think the concept is more general, but it's probably an approach to trying to understand some of the changes that we do start to list, and to detect. So gene variants or alleles are related to different characteristics. Sometimes they are just immaterial, and they don't matter. Most of the cases they don't matter. They're just silent, irrelevant. But some of them do have some effects. And so, in relating to your question, I agree with you that one has to conceive the cognitive niche as something that has evolved itself, with human societies. And of course, Homo erectus was very different from Homo sapiens. 1 million years ago, it was not the same. And even the agricultural revolution created new niches and modified larger societies, modified it. Evolution never ceases. It's impossible for it to cease. It's an intrinsic characteristic of the biological being to evolve. But of course, recently, evolution, I think the cognitive niche is changing so fast, so that there is no possibility for a systematic trend in evolution of the human species. It's not that we are not evolving, it's only that we are evolving like in a Brownian movement, I think. We're not evolving in any particular direction because these processes take many generations. Yet, of course, in historic times, the cognitive niche have changed very dramatically. I'm not sure that it has had an impact on gene frequencies, gene variance frequencies in different populations. That would be the way to study it.
Yeah, I just wanted to make an invitation to everybody in the conference and to Dr. Sobero too, although actually you're involved already, even though you probably don't know it. In one of the big projects we have here in SE3 is essentially associated with obesity as the principal things that we're going to be looking at. And the behavior component, the emotion component is absolutely fundamental. I obviously don't have time to go into that now, but we're working with Samuel Canizales in Meccan, where we're going to be trying to do a genome-wide study of as many people as possible, while we're also looking, we're going to be doing psychological experiments to be looking at cognitive biases. In other words, why do people misperceive, for instance, their weight? Right? So essentially, it's why do people lie to themselves, even though they're actually not lying. They actually believe what they're actually seeing, right? And the thing is, obviously, I think with the Seminario Permanente here as our neighbors, the aspects, the psychological aspects, the emotional aspects are absolutely fundamental, and we would love to have any participation from anybody here who is interested in solving probably what might be the major medical problem of our time, the obesity epidemic.
Very briefly, yeah, I will be very interested in participating, and I agree that it's precisely this emotional and also all other environmental factors that are responsible for the obesity epidemic, because genetics is exactly the same that it was 50 years ago for all practical purposes. So the heritability of obesity diminished with the changes in society. It used to be much more heritable than it is now. So now it's mainly social, emotional. Yet we can conduct a GPS. We can do a GPS and see which many genes, and maybe which is more prone to become obese, and maybe do different, yeah.
Yeah? More questions? No? Todo está.